Hello students, today's class will be oral histology, enamel, second part. And in this class, we will be covering life cycle of ameloblasts, introduction to amelogenesis, enamel proteins, and in brief, we'll be seeing about amelogenesis. Moving on, life cycle of ameloblast. First students, remember if it is amelogenesis or ameloblast, if such terms are used, they are concerned with enamel. Ameloblasts are cells which produce or deposit enamel. The inner enamel epithelial cells of the enamel organ differentiate into ameloblasts which produce enamel. Change in the shape and structure of these inner enamel epithelial cells to become ameloblasts is described as life cycle of ameloblasts. Pay attention because in the past years, in the both last 2 years of NEET paper, life cycle of ameloblast has been an important concept. Now, if you can see in this diagram, you have the inner enamel epithelial cells, which is circled. These cells slowly differentiate into taller cells called as the ameloblasts. All these are ameloblasts, and you can see inner enamel epithelial cells differentiating into ameloblasts. We will see how this differentiation happens. There are 6 stages in the life cycle of an ameloblast: morphogenic stage, organizing stage, formative stage, maturative stage, protective stage, and desmolytic stage. Now, from our exam point of view, how these 6 stages are important is we should know how the ameloblast or the inner enamel epithelial cell looks like in each stage and what happens in each stage. Coming to morphogenic stage, it's the first stage. It corresponds to the early Bell stage. Look towards the image on the right. They are short columnar cells with a large oval nuclei, Golgi apparatus, mitochondria, and centrioles are located at the proximal end surfaces. So if a short cell with a large oval nuclei is given, it is morphogenic stage in the life cycle of an aminoblast. Organizing stage. In this stage, the cell becomes longer and reversal of polarity is seen. What do you mean by reversal of polarity? When the nucleus moves away from the side of the basement membrane. Here, this is the side of the basement membrane. When the nucleus moves away from the basement membrane, it is reversal of polarity. During the terminal phase of the organizing stage, formation of dentin by odontoblast begins. And in this stage, reciprocal induction is seen and reversal of nutrition is also seen. Let me elaborate both. Now, what do they mean by reciprocal induction? It is nothing but the inner enamel epithelial cells interact with the dental papilla and induce odontoblasts to secrete dentin. After the dentin is laid down, the odontoblasts stimulate the inner enamel epithelial cells to differentiate into ameloblasts and later secrete enamel. This is called as reciprocal induction. In short, dentin is laid first, followed by enamel. And what is this reversal of nutrition? In just picturize the enamel, the tooth germ in your mind, students. In the tooth germ, you can see the enamel organ, the dental papilla in the middle of the enamel organ, surrounded by the dental sac. If you can see in this image, this is the enamel organ, dental papilla in the middle, surrounded by the dental sac or the dental follicle. Okay, so during the organizing stage of life cycle of ameloblast, what happens is, so far the nutrition or the blood supply to the inner enamel epithelial cells had been from the dental papilla. But in the organizing stage, after the dentin is laid down, the blood supply to the inner enamel epithelial cells is from the dental follicle or the dental sac. This is called as reversal of nutrition. That is, in short, till before heart tissues are laid down, blood supply or nutrition to the inner enamel epithelial cells is from the dental papilla. After the heart tissues are laid down, that is after heart enamel and dentin and enamel is laid down, nutrition or blood supply to the inner enamel epithelial cells is from the dental sac or follicle. This is seen in the organizing stage of ameloblast life cycle. Formative or secretory stage. Now, if you can see this image, this is the image of the ameloblast in the formative or secretory stage. You can see a horn-like projection here. That is the Tomes process. Tomes process is nothing but extension of the ameloblast into the enamel matrix. Extension of ameloblast into the enamel matrix is called Tomes process. Ameloblasts enter the formative stage after the first layer of dentin has been laid down. Development of Tomes process is characteristic of this stage, and enamel rods and interprismatic substances are formed. Maturative stage. Now in this stage, if you can see, I have given two diagrams. One has a ruffled border or a villi-like border at one end, and the other one is a plain flat, and the other is an image with a plain flat border. In the maturative stage, the ameloblast keeps changing between these two appearances, and that is called as modulation. Development of smooth and ruffle-ended borders in the apical cytoplasm of the cell in cervico-incisal direction keeps changing. Either they are ruffled, sometimes they are ruffled, sometimes they are smooth. And this interchange in the structure of the ameloblast is called as modulation. Modulation happens in the maturative stage of ameloblast life cycle. Here, Tomes process disappears and absorption of proteins and deposition of minerals happen. This was a previously asked MCQ, picture-based MCQ in one of the past NEET papers. They had given a ruffled ended diagram and asked to which stage of ameloblast life cycle this cell belongs. This belongs to the maturative stage. This is called as microvilli. These ruffled borders are called as microvilli. And if microvilli are seen, they belong to the maturative stage. Protective stage. Here the ameloblasts become cuboidal in shape and assume the name of reduced enamel epithelium. They are reduced in size and shape, and they're called as reduced enamel epithelium. No more mitosis of ameloblast takes place. Reduced enamel epithelium protects the matured enamel by separating it from the connective tissue until the tooth erupts. Cementum and radicular dentin formation is seen in the protective stage of ameloblast life cycle, and Hertwig's epithelial root sheath is broken in the protective stage of ameloblast life cycle. Desmolytic stage. The reduced enamel epithelium secretes certain enzymes which destroy the connective tissue and pave way for tooth eruption. This is what the last stage of ameloblast life cycle does. We'll have a short recap. There are 6 stages: morphogenic, organizing, formative, maturative, protective, and desmolytic. Morphogenic stage, large nuclei in the center, short columnar cells. Organizing stage, tall columnar cells, reversal of polarity, reciprocal induction seen, reversal of nutrition, that is from dental papilla to dental sac, reversal of nutrition is seen. Formative or secretory stage, something important to note is the Tomes process. Maturative stage, modulation is seen. M for M, maturative stage will show microvilli. Maturative stage, microvilli. So remember that. Sometimes microvilli may not be seen. Protective stage, the cell decreases in size and becomes reduced enamel epithelium. Desmolytic, the reduced enamel epithelium secretes enzymes and paves way for eruption. Introduction to amelogenesis. Amelogenesis is the secretion or the formation or the deposition of enamel. Amelogenesis is unique in many ways. Ameloblast, that is the cells which secrete enamel, is an epithelial cells. whereas all other secretory cells like cementoblasts, odontoblasts are all ectomesenchymal in origin. Ectomesenchymal in origin means connective tissue origin. Only ameloblast is epithelial in origin. Non-collagenous proteins are also involved in the mineralization of enamel. We'll see about these proteins in detail. Very important from MCQ point of view, so pay attention. Enamel proteins make up the inorganic portion of enamel. They are of two types, namely amelogenins and non-amelogenins. Amelogenins are acidic proteins and amelogenins are non-acidic proteins. Non-amelogenins are acidic proteins. I come again. Two types of enamel proteins, amelogenins, non-amelogenins. Amelogenins are non-acidic proteins. Non-amelogenins are acidic proteins. Enamel proteins aid in the mineralization of enamel. Difference between amelogenins and non-amelogenins. Kindly note down. Amelogenins are non-acidic proteins. They are 25 kDa in weight. They make up 90% of the inorganic component of enamel. They are low molecular weight and hydrophobic. They are rich in proline, histidine, and leucine. They disappear in enamel maturation period. Non-amelogenin. There are three types of non-amelogenin: enamelin or sheathilin, tuftilin, and ameloblastin, amylin or sheathilin. There is a difference between sheathilin and sheathilin. Both are different. Enamelin is also called as sheathilin. Kindly note down the spelling. And ameloblastin is also called as sheathilin or amylin. It constitutes only about 10% of enamel proteins. It is a high molecular weight protein, rich in glycine, aspartic acid, and serine. It remains beyond the enamel maturation period. Amelogenesis can be split into organic matrix formation followed by mineralization. That is, enamel formation and deposition can be split into organic matrix formation and mineralization. In organic matrix formation, in the following sequence, we can see events. First is development of Tombs process. Second one is distal terminal bars. And the third is ameloblasts covering mature enamel. In mineralization begins from the cusp tip to the cervical areas and begins from the dentino-enamel junction to the surface. Regarding amelogenesis, if you know these clearly, it is more than enough. Note down the sequence of events and abnormalities you should know. Any abnormality or interference in the matrix formation phase will result in enamel hypoplasia or amelogenesis imperfecta. And any interference or abnormality in the mineralization phase will result in enamel hypocalcification. Ending this session with MCQs. First one, microvilli are seen in which stage of life cycle of ameloblast? Option A, maturation stage. Option B, morphogenesis stage. Option C, organizing stage. And option D, desmolytic stage. Answer is option A, maturation stage. Like I said before, M for M. Maturation, microvilli. Second question, amelogenins are rich in which of the following amino acids? Options, proline, leucine, and histidine. But the answer is all the above. Option D, all the above is the answer. Amelogenins are rich in proline, leucine, and histidine. Non-amelogenins are rich in glycine, aspartic acid, and serine. That's all for this class. Thank you.